Good. So we are live now. Hello, everyone. I'm, as usual, very happy to introduce our speaker today. I'm particularly happy, actually, because Gul was kind of hesitant to participate to this series because she didn't know exactly whether to support the, maybe I don't know if she wants to say a couple of words more about this or not, because so to support the online or virtual type of forums. But I'm happy that she's here. I mean, it's not surprise since she's studying social behaviors, I guess, but she's also has something to say about that, how our sociability changes with this type of exchanges. So, so about Gul now, so she earned her MD and PhD at Brown University and the MIT. And already since then, so her thesis already is about autism spectrum disorders or Fragile X syndrome. After a PhD thesis, she moved to Stanford to join the Department of Psychiatry. And there she was working on some paradigm shifting work on the neuro circuits underlying social rewarding learning. Since 2014, she's in Baltimore at Johns Hopkins as a now associate professor. So as I already hinted to, Gul is working on social behavior, and particularly she is interested about studying social behaviors in health and disorders. So with that, I finish with my very brief introduction. Again, Gul, we are very glad, or I'm very glad you joined us. So the stage is yours. Now you can share your screen and start with your talk.
Allow, okay. Oh, I need to do this. Okay, is that working now?
That's perfect.
Okay, great. Thank you so much for inviting me. Yeah, as Gaia mentioned, I am a little bit nervous about this virtual format because, as she said, I study social behaviors, and I really do feel there are limitations, especially for people's ability to learn information in this sort of degenerate social realm. However, you know, I think we're all kind of learning, and we'll see how it goes. So I feel a little bit weird because I'm just talking into a void. I'm just looking at my own computer screen. So it's really hard for me to sense whether or not what I'm saying is making sense to people, or people are amused or disappointed or confused. So unfortunately, I won't be able to use my normal social cues to tailor the pace of the talk, but I've given this talk before, so hopefully it won't be too terrible. So today I'm going to tell you about some new work that actually was just accepted for publication yesterday in Neuron on parallel social information processing circuits, which we find to be differentially impacted in autism. So as Gaia mentioned, my lab works on social behaviors, and we study it from a variety of different perspectives, including development and synapses, circuits, and evolution. And we're particularly interested in diseases like autism and Fragile X, where social behaviors are disrupted, and that this is a sort of central feature of the disease. Today I'm going to tell you about how we're going to merge our understanding of two parallel circuits of social information processing, and how that's going to inform our understanding of autism. So parallel processing is something that, you know, we've kind of known exists as an organizational principle in the brain for a number of years now. And probably the best known example is the visual system, where there are these two streams of visual information, the ventral stream and the dorsal stream. And these two types of visual information encode different aspects of the visual stream, so visual system. So the what pathway is really good at fine granular detail, and the dorsal stream is more attuned to contrast and motion. And these are encoded by in the brain in this parallel stream of information processing, where in the retina we have lots of different cell types. Here we're going to focus on just the midget and the parasol cell types, and those cell types send projections to target cells in the lateral geniculate nucleus of the thalamus, and they stay segregated all the way until the visual cortex, where they are separated into the LGN cells, the magnocellular and the parvocellular LGN cells that receive parasol and midget projections from the retina. Okay, and so that information gets stays segregated in the visual cortex. And what this this parallel processing is thought to allow the brain to do is to massively expand the network capabilities of the system. And, Margaret Livingston had this very delightful paper 20 years ago now, sort of postulating that the two visual systems working in parallel might explain why people have this longstanding fascination for the Mona Lisa, for example. So this is a painting by Leonardo da Vinci, and people have been mesmerized by the fact that whenever you look at her mouth, she's not really smiling, but when you look at her eyes, you have this sense that you can never catch her smiling, but you know that she is. And what she proposes is that the M pathway sees this high contrast image, and you can see that in this image, the high contrast shadows suggest a smile, whereas the P pathway sees this fine detail image, and there's no smile in this image. And so when you're focused on the eyes, the P pathway is seeing this, and the M pathway is seeing this. When you're focused on the mouth, the P pathway is seeing this, and the M pathway is seeing the eyes. And so this mismatch between the information that's coming to the two pathways is what's giving this sort of curious smile of the Mona Lisa, is what she proposed in this paper. So we've known for a long time that there are magnocellular and parvocellular oxytocin neurons as well. So this was first described in the 1920s and early 30s. And in the hypothalamus, it was noted that there are these two subtypes of oxytocin neurons that are sort of intermingled with a bunch of other neurons. And so we, some years ago now, postulated that these magnocellular and parvocellular oxytocin neurons might also segregate the social brain into two parallel streams of information processing. And that idea was based on the fact that these cell types seem to be specialized, their cellular functions seem to be specialized, and that these cellular specializations might enable them to do different behaviors. So for example, the magnocellular pathway, these neurons exist in the PDN and the SON of the hypothalamus. They send axonal projections to the posterior pituitary, where oxytocin is released into the systemic circulation. It binds to receptors in the periphery, including the uterus and the breast, and activation of these receptors are responsible for lactation and parturition behavior. And the idea is that because these magnocellular neurons also release a huge amount of oxytocin into the circulating CSF of the brain, that they can coordinate the bonding between mother and baby with the lactation and parturition behavior that's happening systemically. In contrast, the parvocellular pathway doesn't send projections to the posterior pituitary. The neurons are restricted to the central nervous system. And we had some sort of circumstantial evidence that maybe they were sending projections to the nucleus accumbens and working via sort of more localized perisynaptic release mechanisms through medium dense core vesicles. So they're releasing a lot less oxytocin, and they seem to be more directed at specific synapses. And what we propose is that this might be great for encoding more moderated or measured social attachments, such as those formed between peers. Because after all, whereas, you know, it might be really important for a mom to start lactating the minute that she sees her baby, it would probably be disadvantageous if you're playing poker with your friends if you start lactating when you see that, you know, somebody, your best friend, is coming down the hall, right? And similarly, you know, it might be important when you fall in love that, you know, you're able to sort of disregard all of the things that are going to annoy you later, like he never puts the toilet seat down, or, you know, the baby is always crying. And you just like feel like you're in love, everything is roses, and you just like fall head over heels for that person. Whereas when you are making your peer-peer social attachments, you probably want to have less of that sort of bonding, that sort of everything is roses, everything is taken over by that sense of love. And you want to be more measured about it so that you can evaluate whether this peer group member is going to, you know, is going to have your back when you need it, is trustworthy, is able to lend you a hand when you need it. So that was our theory. And it was largely supported by comparative studies across prairie voles and mountain voles and mice and other species. But there was really no direct evidence to support this view. But we also speculated that because autism, social impairments in autism are also domain specific, in other words, that people with autism have no their social behaviors aren't disrupted across the board. They have normal, you know, they're able to form attachments to their parents. They're able to be interested in finding partners and sexual attachments. And it really seems to be restricted, their social impairments, to the peer-peer social attachments or social interactions, that maybe the social deficits that we see in autism might be uniquely encoded by disruptions in the parvocellular pathway. So this was just our hypothesis, mostly indirect evidence to build on this. Okay, so you know, autism is a complex genetic disorder, and we know that in the you know, most people have approached this disease taking a genetics view, but there is this other idea in the clinical literature called Pathoclesis. And what this idea is that it says, look, there are, you know, a lot of different neuronal types, thousands of neuronal subtypes, and that these cell types might be specifically vulnerable to injury. So even though the brain might receive a uniform injury across the whole brain, that some cell types might be more susceptible to those injuries. Okay, and probably my favorite example of this in the clinical literature is Huntington's disease. So here's a disease where it's a genetic disorder, mutant Huntington. The mutations in the Huntington gene lead to these very characteristic movement disorders called Huntington's chorea, which is correlated to a degeneration in the striatum of Huntington's disease patients. Okay, and what has been suggested is that it's possible that this selective impairment or the selective injury of striatal neurons in Huntington's disease reflects the fact that mutant Huntington protein binds selectively or especially well to the RAS homolog enriched in the striatum. It's called RES. And that this would account for why the motor impairments and the degeneration start in the striatum. Okay, and as I mentioned, autism, just like Huntington's disease, is a highly heritable genetic disorder. And to date, we have discovered something like over 1,000 genes that have been identified as risk genes for autism. And most of the data to date, most of the studies that have tried to understand the pathogenesis of autism have really focused on trying to understand the function of these genes and understand how, you know, sort of taking a bottom-up approach, you know, how the function of the disruption of each of these genes could lead to the social impairments that characterize the disorder. And in these efforts, Fragile X has been a really important sort of etiological node, if you will, because FMR1, the gene that is disrupted in Fragile X, binds to roughly 20% of autism susceptibility gene transcripts and regulates them. And so the idea is that if we can understand this sort of network of genes that FMRP is regulating, that we may be able to solve the sort of holy grail of autism pathophysiology, which is understanding how all this etiological heterogeneity is able to produce the clinical homogeneity that defines the disease, because the disease, as you know, is a psychiatric one and is still defined by behavioral impairments, and this is how the diagnosis is made in these patients. Okay, and so despite the fact that FMR1 has this genetic interest in terms of being very important for the etiology, and it's a well established risk gene for ASD. Oxytocin is the evidence supporting oxytocin as a ASD risk gene is a little bit more controversial. Nevertheless, we and others have defined neural circuits, including the hypothalamus, the nucleus accumbens, the VTA, the dorsal raphe nucleus, that make up a circuit of brain regions that are important for encoding social behaviors in mice. And interestingly, of all of those brain regions that have been implicated in regulating sociality in animals, only the hypothalamus shows this exaggerated, very pronounced protein synthesis phenotype that we see in the FMR1 knockout mice. So this little heart-shaped thing is the hypothalamus, and in white, this is showing protein synthesis level using metabolic labeling strategies in vivo, and these are ex vivo images. And in wild types, you see this hypothalamic increase of protein synthesis, and in FMR1 knockouts, this is much more pronounced in the hypothalamus. And we don't see this in either the VTA or the dorsal raphe or the nucleus accumbens. So that we thought was an interesting clue. Okay, so in order to start this analysis to try and understand, to bring together this genetic etiology of autism and the social behavioral circuit, so the bottom-up approach and the top-down approach, we decided that we needed to start by having a more firm handle on this circuit, this parallel circuit that we had defined in our hypothesis, in our review hypothesis from a few years back. And this is because while a lot of the data that we cited was sort of descriptive, really nobody had done the quantitative analysis to describe these populations of neurons. So in order to do this, what we started with was tail vein injections of the blood-brain barrier impermeable tracer Floral into the tail vein. And this gets taken up by magnocellular neurons that send their axonal projections outside the blood-brain barrier, gets taken up to the cell body. Meanwhile, the parvocellular neurons that don't send projections to the posterior pituitary do not get labeled with this Floral. And then we label all of the neurons with the oxytocin antibody that we got from Harold Gaynor. And when we do this, we can quantify magnocellular neurons as being the ones that are oxytocin antibody positive and Floral positive, whereas the parvocellular neurons are the ones that are oxytocin antibody positive and Floral negative. So these neurons. Okay, and when we quantified this, we were able to, just sort of qualitatively draw these out, and this is what the two cell types look like. And when we quantified it, what we found is that in the PVN, 66% of the neurons are, of the oxytocin neurons are magnocellular, and 34% are parvocellular. And what's interesting is that although this had been suggested in the literature, it had never been shown before quantitatively, that the magnocellular neurons reside in the rostral portion of the PVN, whereas the parvocellular neurons tend to cluster in the caudal portion of the PVN. And so This is interesting because it means that, if you're going to be looking for these neurons by retrograde tracing and elsewhere in the brain, it'll be really important to check the whole rostrocaudal axis of the PDN to make sure you're not missing any neurons. So next we wanted to take a molecular genetic approach. So this was enabled by our oxytocin flip knock-in mice that we developed actually in our previous paper that was published last year. And we crossed these oxytocin flip knock-in mice to a flip-dependent GFP reporter mouse, and then cross-validated with oxytocin antibody, and just showed that the antibody neuron labeled oxytocin neurons and the reporter labeled oxytocin neurons are the same. And then we wanted to look at the electrophysiological signature of magnocellular and parvocellular oxytocin neurons. So again, what we did is we took the oxytocin flip GFP reporter mice, and then we injected them with tail vein Floral gold. Then we cut slices and did current count recordings of neurons that were GFP positive. We also filled the pipette with neurobiotin so that after we were done with recording, we could resection the slices, fix them, resection them, and then identify them as being Floral gold positive or negative. So this allowed us to do a quantitative blinded measurement for assessing whether or not electrophysiological signatures could identify magnocellular and parvocellular oxytocin neurons. And this was really important because while Luther and Tasker had developed this electrophysiological signature of magnocellular and parvocellular neurons in general in the hypothalamus, they had never been able to do this in identified oxytocin neurons. So there are other magnocellular and parvocellular neurons in the hypothalamus, including the vasopressin neurons that can be either magnocellular or parvocellular, and then a bunch of other neurons that are parvocellular. So in identified neurons, this had never been done. And so what we find is that if we depolarize the if we hyperpolarize the cells and then depolarize them, in the magnocellular neurons, we get this shoulder before the action potentials start, whereas in the parvocellular cells, the action potentials start right away and there's no shoulder. And this is thought to be dependent on potassium channels. And what we found is that if we quantify the neurons as being shoulder positive versus shoulder negative, that the group that is shoulder positive has a significantly longer latency to first spike compared to shoulder negative. And we get the exact same distribution when we look at Floral gold positive versus Floral gold negative. Same with the duration of the action potential and also the dependence on this potassium channel blocker 4AP. Okay, and so what this told us is that yes, the electrophysiological signature that Luther and Tasker had described for magnocellular and parvocellular neurons in general holds for the oxytocin neurons, but then we wanted to take advantage of the fact that we had a large number of cells and that we could perform quantification on this to determine whether or not these electrophysiological features were enough to predict cellular identity based on those features. And so we used an unsupervised k-means clustering method. And what we showed is that yes, indeed, just by using latency, action potential frequency, and duration, magnocellular and parvocellular oxytocin neurons cluster into two groups, and that they nicely divide between shoulder positive and shoulder negative, and in almost all of the cases, also Floral gold positive and Floral gold negative. Sorry. Okay, so next we wanted to see whether or not we could identify magnocellular and parvocellular oxytocin neuronal transcriptional signatures. And so what had been done previously is that there had been a couple of papers where they had looked at the whole hypothalamus and done single cell sequencing. And in these studies, the capture rate of oxytocin neurons was so low that unfortunately, they were not able to make meaningful distinctions between subtypes of oxytocin neurons because they just didn't capture enough of them. More recently, other studies have tried to improve on this by taking the molecular genetic specification approach to sort of amplified the amount of oxytocin cells in their sample. But in that study they had done, they didn't do single cell sequencing, so they weren't able to really finally look at the subtypes of genes that were in the two cell types. So here we took both of those approaches and we combined that with the projection targeting approach. So we used, again, the oxytocin flip reporter mouse to increase the proportion of oxytocin cells in our sample. We use Floral labeling to use projection target mapping to give us another cross-validating method for identifying magnocellular and parvocellular neurons. We dissected and digested the hypothalamus out. We made a single cell suspension, and then we enriched using FACS sorting for green cells that are presumptive parvocellular neurons, and what are labeled here as purple cells. So these are double labeled between the GFP and the Floral. And we just created a library, and then we did sequencing and analysis to look at these different single cell analysis. And so when we did this single cell analysis, what we found is that there were the best fit for our clusters using uniform manifold approximation and projection, so Umap embedding method. When we did this, we found that the best fit was two clusters, which you can see here mapped out in this flattened multi-dimensional space of the two clusters. And it clearly divides into these two clusters. And so what we wanted to know is, do these two clusters map neatly onto our parvocellular and magnocellular subtypes? So then we took advantage of the fact that we had also Floral labeled these cells. And what we found is that the one of the clusters, the smaller cluster, all of the cells were Floral labeled, whereas none of the Floral positive neurons were found in this cluster. In contrast, in the larger clusters, over 70% of the cells were Floral positive, and suggested that and this sort of mapping allowed us to assign the magnocellular cells to this cluster and the parvocellular cells to this cluster. We also have some other data that I'm not going to show today, suggesting that this is the small number of cells that are Floral negative in this population are probably due to a technical artifact. But in order to cross-validate it yet another way, we took advantage of the fact that in the literature, it's been known that magnocellular and parvocellular oxytocin neurons have different mitochondrial densities. And so magnocellular neurons are mitochondrial rich, and this is thought to enable these neurons to have the energy to make the massive quantities of oxytocin that they need to produce for release through large dense core vesicles, whereas this density is lower in parvocellular neurons that make much smaller amounts of oxytocin. And when we do this analysis, again, what we see is that the parvocellular, that the mitochondrial ratio is matches on to what we had expected based on the previous literature. In other words, there's an increased density in the magnocellular versus parvocellular. And when we cross-check that against the Floral positive and Floral negative, once again, we see that the Floral positive neurons are exclusively in the magnocellular high mitochondrial density group, and the negative ones that the parvocellular group only has Floral negative neurons. And furthermore, the Floral negative neurons are evenly distributed across this. So it's not like all of the Floral negative ones are in this lower cluster, suggesting that they are a subtype of, mixed cell type between these two. So again, consistent with our idea that this is a biological artifact, a technical artifact rather than a biologically separate subset of neurons. Okay, so now we wanted to go and look more deeply and try and understand the genes that are differentially expressed in these two subsets of cells. And so what we found is that we're using our rigorous statistical criteria. We were able to find 181 genes which are differentially expressed in the two cell types. And just to sort of orient you to this, I've highlighted the 4 that we'll sort of focus on for further cross-validation. So in the Parvocellular group, we have the Relin and CNR1 genes. So this is the genes that are differentially expressed in parvocellular neurons, and the KCNB4 and the Calbindin 1 gene that are differentially expressed in the magnocellular neurons. And you can see this in the Umap. So here the Relin is largely clustered in the parvocellular group. The CNR1 is largely clustered in the parvocellular, whereas Calbindin and KCNB4 are clustered in the magnocellular group. And this is just Violin plots showing the same thing. Okay, and then we wanted to cross-validate that again, and this time we decided to do using an RNA, mRNA method. We looked at transcripts of oxytocin and Calbindin, and we wanted to cross-validate this across the rostral-caudal distribution that I had told you about in the first slide, or in the first data slide, where we had shown that in the rostral PVN, the majority of cells are magnocellular, whereas in the caudal PVN, the majority of oxytocin cells are parvocellular. And consistent with our transcriptional profiling, what we found is that the magnocellular discriminatory marker Calbindin co-localizes with oxytocin neurons in the rostral PVN, whereas CNR1 co-localizes with oxytocin positive cells in the caudal PVN. Okay, so I just want to emphasize here, because this is something that has confused some of our reviewers, we are not making the claim that Calbindin and CNR1 are identifying markers of oxytocin, parvocellular and magnocellular oxytocin neurons, because clearly Calbindin is expressed elsewhere in the brain, but amongst oxytocin neurons, it's able to differentially identify magnocellular versus parvocellular neurons. And you'll see why that's going to become important in a second. Okay, so now we wanted to go back to the autism question. So as I mentioned in the introduction, autism is characterized by domain-specific impairments in social behaviors. And to date, everybody basically uses the 3-chambered social approach task, and every study does it slightly differently. Some studies compare the social interaction between an adult and a juvenile, some between 2 adults, some between 2 different strains, sometimes males and females, sometimes just males, so it's all over the place. And we think that it's really important as we move forward in the understanding how to translate animal studies into clinical trials, that we not only improve the face validity of the behavioral tasks that we use in mice, but we also try and replicate different aspects of the social impairments that are seen in humans. And so, as I mentioned, in human studies, there is evidence that the impairments are not only social domain specific, so only in peer-to-peer social interactions, whereas parental attachments, for example, are intact. But when the studies have used really good controls for patient salient objects, that also the domain specificity of the social impairments means that they don't have sort of general impairments in reward learning, people with autism. Okay, so we wanted to test the social motivation theory of autism, which was proposed by Chevalier and colleagues in 2016, I think. And in order to do that, we used an assay that we really like, which is called the social condition place preference task. And this is a task that was actually adapted from 30 years of studies looking at condition place preference for looking at drug reward. And so I'm just going to run through this quickly, because we're going to use this behavioral task for the rest of the behavioral slides I'm going to show you. So the animals start off in a home cage where they're in bedding that's familiar to them, and then they're placed into an experimental chamber where there's 2 novel types of bedding that they've never seen before, and that those 2 beddings are separated by a partition. that has a hole in it, and they can freely pass between the two types of bedding. And this partition, and then we just take a 30-minute preconditioning baseline measurement to make sure that they don't have any preference for one type of bedding or the other on average, okay? And then the animals are placed in one of these two types of beddings with their social group members, and then conditioned to that bedding for 24 hours is the social condition, and then to the other type of bedding for 24 hours by themselves for the isolate conditioning. And then we place them back in the chamber and do another 30-minute post-conditioning trial where we ask how much time they spend in each of the two types of bedding. And if they spend more time in the bedding that they have been conditioned with their social group members, we interpret that to mean that they have a learned preference for a learned association between the bedding and their preferred stimulus, which is the social conditioning. Okay, there are certain other caveats to this, which I'm happy to discuss in the question answer, but we've published this several times before, and it's now sort of an established. We didn't actually invent this. This was invented by Panksepp and Lavis, and we just used it in several other publications. So I'd be happy to take more questions about this if there are any at the end. Okay, so first we just wanted to look at the FMR1 knockout mouse and look and see whether or not they have any impairments in social CPP. So we looked at wild type and FMR1 knockout mice, and up here is the individual animal responses and their pre-responses and their post-responses, and anything below 900, so between 400 and 900, is the time spent in the box that was assigned later to the isolate conditioning, whereas anything in this top part was time spent in the social conditioning. And you can see it in this individual traces, but it's much clearer in the average traces that on average, before they were conditioned to any bedding, they spent approximately equal number of amount of time in each of the two chambers, but after conditioning, they spend significantly more time in the bedding that was conditioned to the social condition. Oh, I'm sorry. What did I do? I moved ahead. So this is the social CPP. I don't know how it jumped ahead on me like that. Sorry about that. In the FMR1 knockout mouse, what we see is that in the preconditioning, again, they don't show any preference, and after conditioning, they show no preference for the social conditioning. And this is just quantified and across groups with normalized social preference and subtracted social preference for them. You can see that there's a statistically significant decrease in the amounts spent in the social condition compared to the isolate condition in FMR1 knockout mice. But if we now go to cocaine condition place preference, so this is a different protocol now than the cartoon I showed you above. This time, instead of conditioning the two types of bedding to social and isolate, now they're being conditioned to cocaine or saline. And when we do this measurement, what we find is that in both the wild types and the FMR1 knockout mice, we see a significant preference for the bedding that is conditioned to the cocaine stimulus, suggesting that the impairments in FMR1 knockout mice are domain specific to a drug reward, just like in human patients with autism, there seems to be no domain general impairments in other rewards, patient salient objects, etc., seen in autistic patients. Similarly, when we change the protocol and instead of making it social versus isolate, now the animals are trained with an allo parent. So this is a sort of a babysitter female that has been put in the cage with the mom, and that's just for technical reasons that I can get into if anybody's interested. Versus isolate, the FMR1 knockout mice also prefer to spend time with the allo parent compared to the isolate. So for the social attachment between the allo parent social attachment, FMR1 knockout mice seem to have no impairment. And again, this is consistent with the human literature showing that that people with autism have no attachment disorders in terms of their attachments to their parents. Okay. Okay, so now returning to our question of the circuits, we wanted to ask whether or not the circumstantial evidence that we had from previous studies that the nucleus accumbens receives a parvocellular oxytocinergic projection could be validated using our electrophysiological criteria that we established in the earlier studies. So here what we did is we took our oxytocin to a flip GFP reporter mice. We injected the nucleus accumbens with retrobeads, and then we cut slices, and we recorded from retrobead positive, GFP positive neurons in the PDN. And then we just looked at their electrophysiological signature using that same current cramp protocol I showed you earlier. And what we found is that by latency to first spike, duration of action potential, and potassium channel dependence, that all 9 of the neurons that we recorded using this method fit in the parvocellular cluster. And we were able to quantitatively validate that using our k-means clustering analysis as well. So that gave us confidence that the accumbens, unlike other brain regions where we have examined this before, that receive a mixed projection of magnocellular and parvocellular oxytocin neurons, for example, in the VTA, which was published in Hung et al. In 2017, and then some unpublished work from the amygdala and medial preoptic area showing that all those other brain regions received a mixed projection. In the nucleus accumbens in male mice receives an exclusively parvocellular oxytocinergic projection. And what this allows us to do is test the requirement of parvocellular oxytocin neurons for these social reward learning impairments in FMR1 knockout mice. So in order to do that, first we had to start by determining whether or not FMR1 is required in an ongoing way in the nucleus accumbens for these social impairments that we see in the constitutive mice. Constitutive knockout mice. So here we use the conditional FMR1 knockout mice. And so this allows us to delete FMR1 in only the cells that are expressing CRE. And we took advantage of the K9 adenovirus that expresses CRE, which we injected bilaterally into the nucleus accumbens. And this virus is a retrograde virus. So it gets picked up by all of the neurons that send projections to the nucleus accumbens. And when it gets transcribed and translated, the neurons that have this virus in it will make CRE and shut off FMR1. And so we'll delete FMR1 in all neurons that send projections to the nucleus accumbens in an acute way. So in adulthood, when they're postnatal day 28 to 35. And when we did this, what we found is that just like in the constitutive knockout mice, if we injected CAV CRE into the conditional FMR1 knockout mice, we saw no social CPP. Whereas if we injected CAV GFP into the conditional FMR1 knockout mice, we saw significant social reward learning. And so this really sort of challenges recent or it suggests that, recently there was a high profile paper from David Ginty's lab showing that, hysteresis might play an important role in the pathogenesis of social impairments. In other words, if you delete genes early on in development, you get the social impairments, but if you do it later in development, you don't get the same impairments. And this really argues against that, or at least it suggests that mechanism is not universal across different etiologies of autism, because here this is clear evidence that ongoing FMRP, the ongoing requirement of FMR1 for social impairments in the FMR1 knockout model of autism. Okay, so next we wanted to sort of zero in and be a little bit more specific. So the K9 adenovirus strategy I just told you about is targeting all of the projections to the nucleus accumbens, but not including the oxytocinergic projections. But there are a lot of other different cell types that send projections to the to the nucleus accumbens besides oxytocin. So now we wanted to look at just those oxytocin neurons. And because we had just shown that the nucleus accumbens receives an exclusively parvocellular oxytocinergic projection, this really sort of gets at the parvocellular, magnocellular question in a circuit specific way. And so here what we did is we used a retrograde AAV that is expressing a flip-dependent Cre injected into the nucleus accumbens in mice that were crossed between a oxytocin to a flip mouse and a conditional FMR1 knockout mouse. So what this is going to do is it's going to knock out FMR1 in only the oxytocin cells that are expressing flip, who then turn on Cre and then knocks out FMR1 in those cells. Okay, so it's sort of our fanciest molecular trick to specify oxytocinergic projections to the nucleus accumbens for FMR1 knockdown. And when we do this, once again, we recapitulate the constitutive knockout phenotype, suggesting that parvocellular oxytocinergic projections to the nucleus accumbens are required for this behavior. And so this, I think, is the first direct demonstration that parvocellular oxytocin neurons, which had been implicated in a bunch of other functions not related to social, are directly responsible for a subtype of social behavior, namely peer-peer social reward learning. Okay, so this was exciting, but it might have been the case that, you know, anytime you muck up any type of oxytocin in the brain, you're going to get an impairment in social reward learning. It's just that important for social reward learning. So maybe it's not specific to parvocellular oxytocin neurons. And if we did the same manipulation in magnocellular neurons, we get the same result. And so next we wanted to look at the Calbindin Cre mice, because as I mentioned, Calbindin is a discriminatory marker for magnocellular neurons. And so what we did is we took the Calbindin Cre mice and crossed them to the conditional FMR1 mice. And so this is going to knock out FMR1 in all of the cells that express Calbindin, including the magnocellular oxytocin neurons and a bunch of other neurons that are not oxytocinergic at all, but none of which are parvocellular oxytocin neurons. And when we did this manipulation, sort of amazingly, actually, I never believed that this was going to work, so I was kind of blown away when it did. The FMR1 knockout mice show no impairment in social reward learning. And this is really important because it says that the effect of the deletion of FMR1 and parvocellular oxytocin neurons is really specific, and that you don't get the same impairment when you delete FMR1 and magnocellular neurons. Furthermore, it sort of suggests that, you know, because Calbindin is highly expressed, say, in the Purkinje cells in the cerebellum, previous reports implicating, you know, cerebellar Purkinje cells in the pathogenesis of social impairments in, say, tuberous sclerosis mice, that finding doesn't really generalize across autism etiologies. Okay, so that was a really important finding for us. Okay, so next, we sort of had this idea that, all right, the holy grail in autism is to find a phenotype and see whether or not that mechanism that you've identified generalizes across a lot of different types of etiology. And one of the things that has been difficult in the last 20 years or so, trying to do this bottom-up approach to finding overlap between these genes is that, you know, in that list of genes I showed you, the 1,000 or so autism risk genes, you know, the genes all do very, very different things. And so finding a way to find the intersection of those genes at the level of biochemistry has been very, very difficult. And so we just started with the assumption that, you know, these genes do different things, but that impairments in them, if they are somehow parvocellular oxytocin neurons are selectively vulnerable to those types of genetic injuries in those genes, then it should show up in the transcriptional profile of the oxytocin, magnocellular and parvocellular oxytocin neurons in the differentially expressed gene list. And so what we did next is to look at the differential. We looked at all genes, then we looked at all ASD risk genes, and then we looked at differentially expressed ASD risk genes across our sample of magnocellular and parvocellular genes. And what we found is that both ASD risk and FMRP binding partners are enriched in the population of all ASD genes, all FMRP binding partners. They're also enriched in the differentially expressed genes for both cases, and in the subset of genes that are intersecting between those two. And so this was remarkable because it really says that the reason that we've been missing the ASD, the involvement of oxytocin in ASD pathogenesis is that, roughly 60% of the oxytocin neurons aren't really involved in the pathogenesis of the disease. In other words, the magnocellular neurons, whereas the parvocellular ones represent a subset of those cells, or a minority subset of those cells. But in those subsets, these genes are very highly expressed. Okay, so to our knowledge, this is the first time that anybody's been able to make this etiological leap across multiple different causes of autism to this extent. Okay, so it might have been that we were just sort of suffering from looking under the lamppost. So we wanted to find autism genes and we wanted to find parvocellular, and that's why this was happening. And that, if we had looked at any disease that has a genetic, there might be something about parvocellular neurons where, genetic diseases, they're just more susceptible to genetic diseases. So we looked at gene sets from a number of other diseases that are not defined by primary deficits in social impairments, including schizophrenia risk genes, Alzheimer's disease risk genes, and type 2 diabetes risk genes. And here we found no enrichment no differential expression and no enrichment in magnocellular and parvocellular oxytocin neuronal subtypes. So that really says that, this is really specific to a disease that is characterized by social impairments. And so I'll just wrap things up, I'm a little bit over, just wrap things up by kind of summarizing our data in this working model of what we think is going on. And so I gave you evidence that there are two parallel streams of social information processing encoded by magnocellular and parvocellular neurons. In the literature, there have been 14 different characteristics of these neurons that have been used to differentiate them. And here I gave you evidence that we provided evidence corroborating 8 of these different characteristics, including 2 that we discovered here, which is the discriminatory gene markers and the ASD gene enrichment that we see in the parvocellular but not magnocellular neurons. And furthermore, we provide evidence that these neurons function to encode different subtypes of social behaviors, so peer-peer attachments in parvocellular neurons, and magnocellular neurons are more for filial attachments, and that these are impaired in our FMR1 knockout mice, and these are not. And furthermore, if we disrupt FMR1 in these cells, we don't get any impairments in social behaviors, whereas if we in peer-peer social reward learning, but if we remove FMR1 from these cells, we do get impairments. Okay, so that's sort of our working model, and this is really important because all of the clinical trials to date that have tried to use intranasal oxytocin as a therapeutic have really probably targeted the magnocellular neurons, and this could be an explanation for why these have failed in clinical trials. And with that, I'm just going to end and acknowledge the people who did the work. So Eastman Lewis, Alejandra Patino, and Genevieve Stein O'Brien were the 3 co-first authors on this paper, and they really worked in unison. So Eastman did the electrophysiology and a lot of the anatomy. Alejandra did some of the behavior and the single cell sequencing. Genevieve did the single cell sequencing and a lot of the computational analysis. We also had help from Beatty and Miriam and Dan and Matt and Cooper. And then we also had help from Ian Dardani and Connie Jiang at UPenn. And then we had reagents from all of these people and funding support from these funders. So with that, I can take a few questions.
Thank you very much, Gul. Very beautiful. So I stopped sharing your screen so that we can come back. So there are 2 questions to start with, and then please write your questions on the bottom. Can you hear me well?
Yes.
So first question is, do nucleus accumbens projecting parvocellular oxytocin neurons have axon collaterals to other brain regions besides nucleus accumbens?
They could. We have not formally tested that yet, but given that, yeah, they could. We just don't know that for sure.
So next one, and then don't be shy, ask more. I mean, I have a few more actually, but so does activation of the parvocellular oxytocin neuron protecting to the, I guess it's projecting to the nuclear accumbens induce CPP?
Wait, say that one more time. I got distracted.
Does activation of the parvocellular oxytocin neuron projecting to the nuclear accumbens induce CPP?
Okay, so we didn't do that experiment in these studies, but what we did do that experiment using optogenetic strategies in our 2019 neuron paper. And there, what we did is we took advantage of the fact that this social reward learning has a critical period. So it peaks in sort of around postnatal day 42 when the animals reach sexual maturities. And then by the time they reach mature adulthood, social reward learning is absent in wild type mice. And so what we did is that when we stimulate the oxytocin neurons, which in that paper we hadn't formally proved it yet, so we were just calling oxytocin neurons projecting to the nucleus accumbens, which were targeted with retrograde, flip-dependent channel rhodopsin proteins. And when we stimulated those axon terminals in the nucleus accumbens, then we were able to bring back social CPP in the adults, and we were able to bring back oxytocin-dependent synaptic plasticity in the adults. So yes, sort of in an indirect way, we were able to show that that induces it, but we haven't done anything like real-time, sort of operant conditioning with these, because for a peptide, I'm not sure that it really makes that much sense. I'm not really sure what advantage you're getting doing it optogenetically, since when you're stimulating these neurons, you're releasing by volume transmission medium dense core vesicles of oxytocin, which is a peptide that kind of stays around for a while. So, so we chose to move away from the acute approach and just look at the ability of to bring back the behavior.
So next question is, the differential expression of reelin seems suggestive. Have you looked at the reelin expression in the parvocellular neurons in the FMR1 knockouts?
Yeah, we thought about it and we tried, but reelin is this really complicated protein, which in early development is really important for determining the position of the neuron in layers in the cortex, but in adulthood or postnatally, it actually acts as an extracellular matrix protein. And so when we just try to do antibody labeling, we just saw it all over the extracellular matrix, and it made it really difficult to understand whether it was related to the cell that we were caring about directly, or whether it was having some sort of network effect. And so we really just weren't able to make a clear conclusion based on that. We are sort of interested in the cannabinoid receptor one, which is also sort of interesting that that's in the parvocellulars. And, I like to, speculate that might have something to do with some of this social anxiety that people report when they, do use too much cannabis, for example. So maybe, I don't know, we haven't done anything related to that, but it's possible that that's an important part of that phenotype.
So next question, are attachment-related conditioning measures harder to extinguish than peer-related?
Sorry, I say it again.
If the attachment-related conditioning is harder to extinguish compared to the peer-related.
Ooh, yeah, no, we haven't done anything like that. I mean, it's a good question. You know, it would be nice to really fully characterize this, but we will really, this is the first time we've ever used it, and so we're gonna, you know, I think now that we have this as a way to test the different arms of the tree, we really want to go to town and really understand what are the other differences, and possibly, I mean, I would love to identify a subset of, you know, if we could find some magnocellular only neurons and show that they were really disrupting those would disrupt that alloparenting behavior. I think that would, you know, really put us on good footing, and we have a lot more work to do on that. So, I mean, I actually think that, like, there are so many questions that this study opens up. You know, I think that we want to start looking at development. We want to start looking at inputs and outputs. We want to look at, you know, there's a lot of other things, and we are not at all territorial about this. So, because there's just not enough time in any one person's lifetime to look at all of this.
That's what they say always as well.
So I would love it if people would take this up and really go to town, because there's a lot here, and, you know, it took people in the visual system, and they're still working on it, you know, 20 years to figure out the parallel circuits. And obviously this is a heuristic, so there might be some subtleties that are, you know, not captured by this dichotomizing of the behaviors. And, you know, this is a good starting point. And what's nice is that I think, you know, we've got now the tools where we can sort of get at these questions, right? So we can use multiple feature selection. So we can combine the oxytocin flip mouse with a Calbindin mouse or with a CNR1 mouse, and then start to specify these subsets of neurons, and then use those enrichment protocols to discover really selective, not just discriminatory markers between the cell types, et cetera. And we can do it across development. So, you know, I think there's a lot to be done, and this work really lays the technical foundations for being able to do that work.
I have just two very quick, probably, questions. One is about your social behavior task. So you have used alloparents or unfamiliar mouse or mice. What if you use not parents but familiar, so that you kind of raise them for a certain time, so they have been meeting those mice before? In your task?
So we used alloparent versus nothing. So either by themselves or getting to be with the alloparent.
Right.
So we never did the unfamiliar familiar. It's a good question. We could certainly do it. The reason that we chose alloparent is because we knew that Barbara Thompson was developing the social CPP task for humans. And she basically used a sort of nurturing female maternal type as the social signal. And she was an adult. She was actually the investigator, right? And so we wanted to map it as closely to that as possible, as somebody that the subject would associate with a adult sort of caregiver type role. And what Barbara Thompson found is exactly what we found, which is that the autism patients, you know, showed no impairment in their social reward learning towards that alloparent figure. And so, yeah, there's a world of things to do, and that would certainly be interesting to know how much this is about the specific social interaction and how much it's about novelty. Because I've often speculated that, you know, one of the major differences between three-chambered social approach and the social CPP task is that, you know, what's being measured in three-chambered social three-chambered social approach task is really social saliency or some feature of novelty. Exactly. And we are trying to stay away from measuring novelty because there might be additional novelty aversion phenotypes in autism that are unrelated to the social.
Exactly. And then the very last, very quick question about your enrichment of ASD-related genes or also FMRP target genes that you find in the parvocellular cell. Whether there is any specific, whether they are converging along any specific function or biological properties, location, anything.
No, I mean, we actually hoped, we did an analysis, just sort of a quick and dirty, where we looked at the differentially expressed genes that, based on some gene ontology stuff, might be expected to be related to electrophysiological properties. We found no specific correlation. The genes are really all over the place and sort of not ones that we would have picked if we were curating a list by any stretch of the imagination. So, I mean, I think that this is exciting because it means that, like that just doing gene ontology mapping isn't going to give us the answers, that we're going to have to stop deferring all of the etiological considerations of these diseases to geneticists alone, right? Like we have to start combining the genetics approach with the circuit mapping approach, or we're going to miss things because, they're just not obvious based on what we know about what the genes do. I also would say that, we focused on one specific time window for the transcriptional analysis because we wanted it to be as closely matched to the time window to the social behavioral assay that we were going to be using and the electrophysiology and everything else that we did. But we think that this is a conservative list and that there are going to be many more genes, probably at the level of transcription factors that are, turned on early in development and turned off by the time we did this analysis, that are also going to enable us to differentially, look across differential expression. And we think that will be important for autism because we also know that the other major node for autism genes is the CHD8 gene, where it's chromatin remodeling kind of thing, and it's early in development and it's basically turned off by the time they're in postnatal development. So we think there will be different, and maybe it will be that if we look early in development, the differential gene expression stuff will be all on the magnocellular, and that there will be hysteresis and there will be an important role for the cerebellum. We don't know, but so I just want to make sure that it's clear that we're talking about a very specific snapshot of this time window, and in these genes, it's a conservative list, but
identify that time window and what type or subtype belongs to that time window is specifically particularly important because of the clinical trials.
Absolutely, yeah, that's right. Because we're not going to be able to intervene anytime soon for those earlier windows. So it's really important that we focus, try and find out.
Fantastic. So I think we have to finish here. I would have other 10 questions, but maybe I write to you later. And was really nice, and congratulations for acceptance of the paper. And thank you again for accepting this invitation. I hope that at the end was not such a bad experience.
No, it was okay. I muddled through. I hope that the audience was able to not feel like I was just talking at nothing. So,
fantastic.
All right. Okay, great.
Thank you so much again.
Thank you. Bye-bye.
Bye.